Okay, thank you very much. A very good morning to all of you. And I'm indeed delighted and honored to be called upon to give this talk, which is essentially on cancer genomics. It's by, the inferences are based on analysis of large-scale genomic data. So I'm going to start with my talk with two stories. It might sound a bit morbid in the morning, but I can assure you that the sun will shine at the end of my talk. So let me start with the two stories that I have. This is my first story. This story comes from the pages of a journal, the New England Journal of Medicine, and that's an individual. This is the whole body scan of an individual, and as we can see, each dot represents a tumor, and as we can see that the cancer in this individual has spread all over the body, and the issue is how do we deal with this individual. So the individual went in therapy, and in a short while, in about 10 or 15 days, this is what happened to this individual. The individual got transformed. As you can see, all of the tumors have regressed from the body, and the individual looks like a normal human being, as all of us would be before the onset of the disease in this individual. However, this is not what all of the individuals face or encounter after therapy. Sometimes even in spite of the same therapy, the individual remains exactly at the same state, and the therapy does not work. So the question is, what is it that makes this individual smile, and what is it that makes this individual sad after therapy? So that's my story number one, and I'll come back to this story as I move along. My story number two is a personal encounter. And the setting is that a person has been admitted to a hospital with lung cancer. I happen to know the director of the hospital. The wife lives far away from the hospital, and this is the conversation that's going on between the oncologist, the cancer clinician, and the wife of the individual who has been admitted to the hospital. So this is the conversation that I've overheard on the hallway of the hospital. The doctor says, well, the patient has non-small cell lung carcinoma. Don't worry about what non-small cell is. Has lung cancer. I need to put him on chemotherapy. The wife asks, can the chemotherapy be done at home? And you know what the immediate reaction of the doctor will be? No. Then he says, wait a minute, let's check out his EGFR mutation status. And I'll come back to what EGFR is. It's epidermal growth factor receptor, and I'll tell you why the doctor said what he said. Then he pauses and says that if it turns out that your husband has epidermal growth factor receptor mutations, then you can take him home and administer chemotherapy orally. And we'll come back to this story again at the end of my talk. Cancer is a disease of the genome, and essentially cancer cells are very disorganized. The hallmark of cancer is unregulated cell growth. If you look at the evolution of the cancer genome, this is a very neatly stacked set of cells. Cancer usually involves multiple mutations, all of these cells, so that's the epithelial layer, and all of these are, let's say, epithelial cells that are all very neatly stacked at birth. All the cells look the same. I'm lying a little bit. They don't all look the same because there are germline mutations that are inherited at the time of birth. But these mutations are kind of innocuous, and as life course of this individual goes on, more and more mutations accumulate. Again, these mutations are innocuous. They cause no harm to the individual. There are some mutations that will give you a slight growth advantage to the cell in which this mutation has happened, but these are benign mutations. Again, these small kinks and bumps do nothing to the individual, and so these kinds of things keep happening. And then there arises a mutation, that's a red mutation that has just appeared, and this mutation gives a huge growth advantage to this cell. It breaks the epithelial layer, invades the inner tissue, and this is what we call as a malignancy. So what we really want to do is to catch hold of these red mutations. They are called driver mutations because they give these cells a huge growth advantage, and that's what we want to do using big data and using genomic sequencing. Again, at the background of all of this, other mutations come along as the life course of this individuals go on. So the major challenge is to generate the data and to be able to identify these red mutations from the background of all of those other mutations that are innocuous, that are really unrelated to cancer, but that's just a biological process that goes on. So the question is, what are the genes and genomic alterations that drive? Essentially, the driving is defined as a growth advantage to the cells. There are lots of people who are doing research on different kinds of cancers, different types of cancers. We've been engaged in cancer that's very prevalent in India, which is oral cancer of a particular type that invades the epithelial cell lining of the oral cavity. So essentially, like I said, those are the driver mutations, and that's what we are after. The squamous cell carcinoma of the oral cavity, let me present to you some epidemiological features. It's the 8th most common cancer. About 250,000 cases arise, new cases arise annually. 2/3 of these arise in developing countries. About 125,000 deaths occur annually. It accounts for about 1/3 of all tobacco-related cancers in India, and between 20 and 60% of all cancer of the oropharynx is associated with the human papilloma virus, which is the most potent biological carcinogen that we now know. The hazardous combination is, of course, the areca nut tobacco on a betel leaf, which is plant-derived. So those are the two major culprits that are oftentimes chewed and kept inside of the lips for the tobacco carcinogens to act upon. Information on patients. Most of our patients are male. It's predominantly a male cancer. 88% of the patients were male. Most were middle-aged. It occurs between 40 and 55 years of age. About 86% presented very late. Most of them are late presenters when it's almost impossible to treat them. Most were exposed to tobacco. 99% of them were exposed to tobacco, more than 80% for over 10 years, and this is mostly smokeless chewable tobacco. About 1/4 of them harbored human papillomavirus infection. So what we did was to do EXOME and whole genome resequencing on about 200 individuals. Essentially, because we are trying to track these somatic mutations that occur during the life course of this individual, we have to sort of subtract out the germline mutations. So we look at the tumor DNA sequence that comprises both the somatic and the germline mutations. Then we look at blood, we sequence those, and that predominantly contains the somatic mutations, and we subtract, so to say, the entire catalog of mutations in the tumor DNA. From that, we subtract the catalog of mutations from the blood DNA to get hold of the somatic mutations. If you look at, so this is real data, and that's big data because it's whole genome sequencing and exome sequencing on about 200 individuals, and essentially I'm going to present to you a summary, not go through the entire steps of the analysis. The number of single nucleotide variants that we find is highly variable across patients. So the x-axis are the patients and the y-axis are the number of mutations, and as you can see, it's sorted. So as you can see that there are some individuals who have very small number of mutations, and there are individuals who have very large number of mutations. What we need to do, as I said, is to be able to sort out the driver mutations from the background mutations, and to be able to do that, one needs to estimate mutation rates, and mutation rate, as we know, is not constant across different regions of the human genome. So what we need to do is to be able to look at a specific region of the human genome and be able to estimate the background mutation rate, the number of mutations that occur in the tumor DNA, and establish that statistically there is a significant occurrence of mutations in that specific segment of the DNA. To be able to do that, since we have to estimate mutation rates for this purpose, and mutation rates are not so easy to estimate, the direct measurement of mutation rate is difficult. So what we do is to use DNA replication time as a covariate using a surrogate of the mutation rate. So essentially, high mutation rate is associated with late replication time and low expression levels. And so what we do is, now there's good data available on replication times and expression levels at various regions of the human genome. We use those as surrogates in order to be able to calibrate whether a specific region of the human genome that you observe in tumors has there is an overdosage of mutations in that particular region compared to the background mutation rate. I'm not getting into the statistical methods because there are various kinds of statistical techniques that are used in order to be able to say whether or not a specific region has a high mutation rate, and so we are able to identify drivers. We also have to do recurrence testing in the sense that we get hold of a discovery from a whole bunch of patients, and then we say that these are the driver mutations, these are the driver genes, and we need to validate those on an independent set, and that's called recurrence testing, and we have actually validated our inferences on an independent set of 110 patients. So what are the genes that drive, and what's the nature of these genes? The genes that drive, well, there are two classes of genes that drive, that are involved with cancers. One class of genes are called tumor suppressors. They are expressed, and when they express normal growth inhibiting proteins, and when there is mutation, they fail to express those, and that leads to tumorigenesis. And then there is another class of genes that are called oncogenes that are usually not expressed, but when they mutate and start expressing, they express these proteins that provide a growth advantage to the cells and do other kinds of things to the cells that make the cell enter the process of tumorigenesis. So in order for cancer to occur, either the tumor suppressor genes have to be obliterated, or the oncogenes have to be activated. So those are one of two ways in which it happens. In terms of oral cancer, what we've established is that it is essentially tumor suppressors that drive. And the tumor suppressors are the usual kind of, many of them are known, such as P53 that occurs in many types of cancers, caspase-8. What we found is FAT1 associated with oral cancer and several other genes, but by and large, the overall impression that we have is that for oral cancers, it's primarily the involvement of tumor suppressors that drive oral cancers. If we look at the entire repertoire of mutations in these various kinds of cancers for each individual and ask ourselves, can we group the individuals by the nature of the mutations or the mutations in the genes in which these appear? And essentially what we found is that there are, of all the oral cancer patients, you can group them into three classes. One class, that's the leftmost one, is essentially driven by FAT1 and caspase-8. The middlemost one, the middle segment is essentially driven by P53, and the third one, the third cluster of individuals, oral cancer individuals, they are driven by multiple heterogeneous mixture of various kinds of genes. So essentially what it is that there are two subclasses, even though it's heterogeneous, there are two subclasses of individuals, one that's predominantly driven by mutations in P53, the other predominantly driven by mutations in caspase-8 and or FAT1. If we look at these subclasses, these vertical bars below, you have to read them the other way around. That is, it's starting from there and going down. Those are the survival times post treatment survival times, disease-free survival times, and as you can see, depending on which particular segment they belong to or which particular subcluster they belong to, the survival times are also quite different. So not only do we have segregation by the gene that's mutated in order to cause the cancer, but also within these larger groups, there are other subtypes that impinge on survival time after tumor. So if we look at one particular one, which is MLL4, patients with mutations in MLL4 have longer disease-free survival. The gain in survival time, disease-free survival time, is about 7 months. 7 months may not seem like a big time, but for cancer relatives or for the patient herself or himself, this is a big deal. 7 months of disease-free survival time is a big deal. We've published all of this a couple of years ago in Nature Communications, and all of the data are presented in that particular paper that we've published. If you look at, so I said that there are mutations, and these mutations provide a provide a survival advantage to the individuals. So if you look at another gene called BRAF, the mutations in BRAF are found in malignant melanoma and other cancers. It's found in 70% of malignant melanoma, 30% of thyroid cancer, 10% of colorectal cancer. The BRAF protein is involved in sending signals inside the cells which are directing cell growth. So essentially it's a protein that directs cell growth, and as I mentioned, that cancer is essentially a problem of cellular growth. So there are mutations that provide a huge growth advantage, and the BRAF protein, when mutated, provides this kind of a problem. So there is a common mutation that occurs in this particular gene, which is an amino acid altering mutation. mutation, and this mutation, BRAF is an oncogene, and this particular mutation provides a growth advantage to the cell when it's mutated. There is an inhibitor called vemurafenib. As I said, that this is a specific that inhibits the protein if this particular amino acid change is present at the 600 position of the amino acid chain. So if you look at this picture that I began with, this individual can either go to this state or to this state depending on the therapy, and it's the same therapy that's being administered. It's not the therapy that counts, it's what happens to this individual when the cancer had occurred. So this individual, this particular individual had mutations in BRAF, and that's a common mutation, was given this particular therapy called vemurafenib, and the individual actually completely recovered. On the other hand, this particular patient did not have the BRAF mutation, and therefore the vemurafenib did not act on this individual, and the individual remained exactly in the same state that the individual started with. So essentially, by analyzing big data and by looking at what kind of mutations are taking place, and if you can find inhibitors, you actually can tailor the therapy to these individuals based on the kind of mutations that you see. As I said, in our case, it's mostly tumor suppressors that drive, and tumor suppressors is bad news because here is a protein that was active, that was being expressed. A mutation has stopped the expression. It's very difficult to recover the expression of that particular protein that has stopped being expressed. So as I also said that the p53 is a major driver of oral cancer, and if we plot the mutations and the genes in which these mutations occur, it has a very long tail of the distribution. Why are we bothered about the long tail of the distribution? Precisely because these tumor suppressors are very difficult to act upon. So the question that we asked ourselves is that if we look at correlations between p53 and other mutations that occur in a subset of individuals, if we plot these on biological pathways, the ones that do not recur in a big way, if we plot them on biological pathways, do we find one particular biological pathway enriched with these mutations? Again, we did that using various kinds of things, and what we find is that the arachidonic acid metabolism pathway is enriched by these small mutations, or the low frequency mutations, and this is the particular pathway, or those mutations map onto this particular pathway, and this particular pathway is enriched in spite of the fact that p53 is the major driver, but these are correlated. So the question is that could we think of an alternative treatment strategy which is to block this particular pathway, and can we block this particular pathway? We were actually quite happy that the arachidonic acid pathway popped up, because there are these COX and LOX inhibitors, and the COX and LOX mutations are associated with proliferation, anti-apoptosis, angiogenesis, and invasion or metastasis. So the COX and LOX genes are good news because there are COX and LOX inhibitors already available in the market. So we are now working with clinicians in order to find out whether we can actually use these as a treatment strategy for oral cancer. Again, with respect to this one, there is a huge growth advantage if there is a mutation in the arachidonic acid metabolism pathway versus those without, and the gain in survival time is about 17 months, which is huge in terms of survival advantage post-treatment. Again, we've published this in Nature Communications last year. One of the next questions that we asked is, all of our patients present themselves at late stages, but supposing we do a population screening for these cancers, are there precancerous lesions in the mouth, in the oral cavity that we can target ahead of time and provide counseling to these individuals? Indeed, there are common precancerous conditions of the oral cavity called leukoplakia, which is the earliest, to erythroplakia, and eventually to oral submucous fibrosis. When the individual comes down with oral submucous fibrosis, the jaws of the individual will not open. So that's almost a pretty late stage. And these are gory pictures, but that's what happens. About 25% of the oral leukoplakia cases proceed to oral cancer. So we thought that maybe we can identify certain kinds of mutations and if we can do a population screening for leukoplakia. So the question that we are asking is, do you find sequential acquisition of mutations as one finds in, for example, colorectal cancer? Again, I don't really have the time to go through all of the various steps. What we found was that there are genes that are significantly mutated in leukoplakia, which is one of the early precancerous lesions, and those are the two genes that are mutated. And then what we found was that even in the precancerous lesion, there are genes that are significantly mutated in there are genes that are common to oral cancer and are significantly mutated even in the precancerous lesion, and these are the three genes that are mutated both in the precancer and in the frank oral cancer. So this is interesting because then it means that there is or at least there are indications of a sequential acquisition of mutations in genes, and therefore we could perhaps interrogate those genes ahead of time if we can find that an individual has precancerous lesions in the oral cavity. The pathway that's significantly altered is the OXPHOS pathway. The oxidative phosphorylation pathway is again interesting from a mitochondrial and other metabolic conditions that are involved with oral cancer. But again, like I said, I don't have the time to get into the details of this. I've shown you this individual, and this individual, as I've shown you, has metastasis all over the body, started with a primary, and then the cancer moved and metastasized in various parts of the body. So cancer metastasis is a major problem, and in oral cancer, the metastasis that takes place early on is a near node metastasis. So there's a lymph node metastasis that frequently occurs in oral cancer patients, and one of the questions that we asked is, can we identify genes that lead to this metastatic process, at least to the near node lymph node metastasis? And what we find is that the BRCA2 is absolutely very strongly related with lymph node metastasis, and if we look at mutations in BRCA2 and grade them by lymph node positive and lymph node negative, what we find is that there is a major difference in survival advantage based on whether or not the BRCA2 gene is or is not altered. And this is so BRCA2 associated with lymph node metastasis again is a prognostic marker as to whether or not the individual is going to develop lymph node metastasis. So finally, I recall the conversation that we have overheard, that I had overheard, and this is the patient has lung cancer. Can the chemotherapy be done at home? No, we need to get his epidermal growth factor receptor sequenced, and then if there are mutations, then we can administer therapy orally. What gives the doctor the confidence that if there are mutations, he can do all of this? The confidence stems from this fact that the epidermal growth factor receptor is a family of receptors that's overexpressed in the cells of the lung and breast cancers. It eventually leads to uncontrolled cell proliferation, and there are mutational hotspots in this particular protein. And there are multiple domains in that particular protein in epidermal growth factor receptor, and the driver mutations are sort of clustered in one particular domain. The mutations in the epidermal growth factor receptor tyrosine kinase domain is responsible for activating anti-apoptotic pathways, and this is known. So one of the studies that was done is to look at a specific inhibitor which is bioavailable, that's called Afatinib, and what was seen is the following, that the standard therapy for chemotherapy for these kinds of cancers is cisplatinum, which is an injectable chemotherapy therapeutic agent, and for cisplatinum the response rate for lung cancer is 23%, but when there is mutation in the epidermal growth factor receptor, one can use Afatinib, which is orally bioavailable, and the response rate is 56%. The progression-free survival is also much better with the oral. It's 11.1 years, 1 month after treatment for the oral chemotherapy, which is the Afatinib, versus the injectable chemotherapy, which is cisplatinum. So where it is about 7 months. So you not only get a bigger response rate, you also get a better survival advantage if you have mutations in the epidermal growth factor receptor gene, where these mutations will allow usage of Afatinib, and that becomes orally bioavailable, and therefore it's a useful oral chemotherapy. So that's why I said that the sun is going to shine at the end. So I've shown you two things. One is using the power of genomics to identify that there are specific mutations that can actually be inhibited, and also you can use not only treatment but also make it bioavailable in less invasive ways, which is making it orally bioavailable. So the dream is to use the power of genomics and big data to turn cancer into a chronic disease, where in the morning, if you have cancer, you just pop a pill in the mouth and go about your daily chores. And to be able to do that would be a huge advantage, and all of us dream to use the power of genomics and big data to be able to do that. Thank you very much for your attention.